Ray asked me to give a very general talk because of the wide variety of interests of people in the audience, and so I'm going to talk about my longest standing scientific interest, which has been episodic disorders. And this started during my residency in neurology at the University of Utah. And the most common episodic disorders we see in the neurology clinic, of course, are migraine and epilepsy, but there are many others, including periodic paralysis, cardiac dysrhythmias, episodic ataxia, and other episodic movement disorders like the paroxysmal dyskinesias, which I'll have more to say about. And although these disorders on the surface are quite different, pounding unilateral headache that puts you to bed, falling to the ground and convulsing, falling over from a cardiac arrhythmia, or having a funny movement disorder, they really, I would argue, and as I've seen more patients throughout the last 20-some years now, they share a lot of similarities, which led me to draw years ago this Venn diagram showing the intersection of these seemingly very different disorders, but there are really lots of similarities which suggested that they had clinical similarities, and I argued back then that this might suggest they shared some common molecular pathophysiology. So I don't have time to go into great detail, but all of these disorders by my selection, are episodic disorders and affect people who between attacks are often completely normal. So this is quite remarkable. The system is working normally much of the time. These people take out their garbage and they pay their taxes. And yet something pushes them over the edge, some precipitating factor. Shown here, the most common stress, fatigue, whether that's just stress or not, I can't tell you. Certain dietary factors, and in some of these disorders, alcohol and caffeine very specifically can induce attacks, as I'll show you. And so this is a similarity among many of these disorders. The response to drugs is very similar. Carbonic anhydrase inhibitors benefit patients from many of these different categories. Tegretol, a great anti-seizure drug, is like a silver bullet at very low doses for patients with paroxysmal kinesiogenic dyskinesia, and on and on. I won't bore you with a long list of these interesting overlaps. Hormonal factors are also quite interesting. These disorders all tend to come on during childhood. They tend to worsen through adolescence, and I presume that this is hormonal, although to be fair, no one has ever proven that. They worsen through adolescence and young adult life, and remarkably, unlike so many disorders, they tend to get better as people get older. There are many forms of epilepsy that tend to get better. Migraine tends to get better as people age. The muscle diseases, the paroxysmal dyskinesias, and the episodic ataxia, as I showed earlier, all of these disorders tend to get better as people get older. And so stress is really quite interesting, and I don't understand, nobody understands what it is about stress, but this is an interesting question that I just want you to keep in the back of your head. Stress is uniformly a precipitant for seizures, headache, movement disorder, cardiac arrhythmia in patients with epilepsy, migraine, whatever order I said those things, long QT syndrome, and episodic ataxias, for example. Now I'd like to refer to this quote that I love so well from William Harvey way back in 1657. "Nature is nowhere accustomed more openly to display her secret mysteries than in cases where she shows traces of her workings apart from the beaten path. Nor is there any better way," and this is why we do genetics, Mendelian genetics, "any better way to advance the proper practice of medicine than to give our minds to the discovery of the usual law of nature by careful investigation of cases of rarer forms of disease." So think about everybody working in Alzheimer's disease pathophysiology, studying proteins encoded by genes that were discovered by studying rare Mendelian families with Alzheimer's. Same is true for Parkinson's. And I'll show you here today that we've learned a lot from studying very rare episodic disorders. And this is the paradigm that is known to many of you, and that was really pioneered in humans by my mentor, Ray White, who I think of as the father of modern human genetics. and that is to study families with strong genetic variants that cause Mendelian, autosomal dominant, autosomal recessive forms of genetic diseases, as I'll talk about today, or disorders, and behavioral variants, not diseases, as Dr. Fu will speak about tomorrow. And to get DNA from these families and to use the DNA and the phenotypic or clinical information from these families to map the genes that are responsible for the phenotype that we see in the family. The gene, as you know, is a blueprint for building the proteins in the cells of our body. We can then study these proteins in test tubes, in cells growing in culture, in animal models, fruit flies and mice. If it's a novel protein of unknown function, to try to understand what the normal protein does, but then also to understand what the genetic variant in the patients or in the sleep variants, for example, how those genetic mutations lead to the phenotype that we're observing in the families. And so all of the examples I show you today are ones that begin with human families, that we've studied the DNA, moved then toward the gene, the protein, structure, function, and then back toward the pathophysiology, and ultimately, we hope such information will lead to better treatments. So I'd like to start now by focusing on the periodic paralyses. I met a woman with hyperkalemic periodic paralysis during my residency and began to study her family, and I decided to set out to try to identify the gene that causes this disorder. So what's the problem here? Genetic linkage mapping, the idea was not novel. That linkage mapping had been done in phage and Drosophila and other organisms, but no one had done it in humans because genetic maps weren't available, and that's what changed when Ray White came on the scene. But the problem is the human genome is huge, 23 pairs of chromosomes, 3 billion base pairs. And so genetic mapping is like walking into a library, a huge library, and the mutation might be a deletion. It might be a page ripped out of one book in the library. It might be a single missense mutation, a spelling error, one letter misspelled in one book in the entire library. And so genetic linkage mapping, first of all, helps us to figure out which chromosome the gene is on, which might be the section of the library that it's in, fiction, say, then what big shelf it's on, and then narrowing in, closing in as much as we can, ultimately, hopefully, to one single shelf containing maybe 10, maybe 20, maybe 100 or 200 books. And then we have to speed read those books or sequence them or use some other strategy for finding the missing page, the spelling error, and identifying the mutation to help us prove that that's the gene. So this is a daunting task. It used to be even more daunting than it is today because of the changing technologies. So this is the kind of family that I was able to collect in Salt Lake City, Utah. Part of the family lives in southern Idaho. But this was the first family described in 1951 by Frank Tyler that has the disorder hyperkalemic periodic paralysis. I saw all 100 individuals from whom I was able to get DNA, 40 of whom were affected by this rare disease. And for the medical students in the audience, if you see 40 people with a really rare disease, you suddenly, even if you're still a resident, you're suddenly a world expert. So that's kind of fun. And it was with this family that I was able to identify or to localize the gene for hyperkalemic to chromosome 7, narrow it to a smaller region, and then the race was on to try to find the gene. The second disease I studied, which turned out to be an allelic disorder, was in a smaller family, but still a pretty respectable family with paramyotonia congenita. This is a temperature-sensitive mutant in humans. These individuals, when their muscles get cold, if they get into a swimming pool, and after their muscles cool down a degree or two, after maybe half an hour or an hour, they will first of all get stiff, and then their muscles will become paralyzed. And a young woman, this young woman here, when she was a young girl, was in a swimming pool and almost drowned, and would have had her father not been there to pull her out when she started crying that she was unable to swim anymore because she had become paralyzed from her muscles cooling because she had been in the water for a long time. So with these kinds of large families, it's possible to map the gene, the causative gene for either of these two muscle diseases. This one mapped to the same place where the hyperkalemic did, suggesting it was the same gene, and in fact it turned out to be the same gene, but a different mutation causing a different phenotype. So then how do we prove, once we've done that, how do we prove that a gene is causative or a mutation is causative for the phenotype that we're studying? Well, first of all, the mutation has to segregate with the disease. All the affected people should have the mutation. It should not be present in controls. If the mutation is present in the general population, then it can't be the cause of a rare disease, because then the people in the general population should have the rare disease, and then they wouldn't be the control population. And then, of course, if we collect multiple families, we expect that we should be able to find multiple mutations in different, mutations in the same gene in different families with the same disease. And this helps bolster the argument that that is really the gene, and that these different mutations are causative mutations in that gene for that disease. So without showing you all of the data, we began marching through. Now, among the periodic paralyses, there are a whole bunch of different phenotypes. The periodic paralyses and non-dystrophic myotonias. These are all similar but clinically distinguishable phenotypes that I don't have time to go into the details, but for the neurologists here, and especially for the neuromuscular people with the neuromuscular interest, these are really interesting phenotypes that are all distinguishable. And we systematically began mapping and cloning all of these genes and showed that all of them, all of these disorders result from mutations in ion channels, sodium channels, calcium channel, chloride channel, and a potassium channel in Anderson-Tawil syndrome. Interestingly, and not surprisingly, as in some other genetic disorders, 4 different phenotypes can result from different mutations in this one sodium channel gene. The first 2 families I showed you were this one, hyperkalemic periodic paralysis and paramyotonia congenita, but also 2 different phenotypes that I haven't shown you families for. And then interestingly, this phenotype, hypokalemic periodic paralysis, different families with an identical phenotype can result, 90% of them have mutations in this calcium channel gene that I cloned 3 years later, but 10% of them have mutations in the sodium channel gene. So different mutations in the same gene can cause different phenotypes. Mutations in 2 different genes can cause the same phenotype. And so this led to completely reorganizing the way we think about these diseases. Initially, we thought about them only clinically, which is still an important way. I don't mean to belittle the way I still think about these clinical disorders, but we now also have to subcategorize them based on the molecular variants that we find and know to be causative of these different disorders. Now, with the exception of the chloride channel, which is an anion channel and completely different, all of these cation channels are evolutionarily related. Unless, of course, you're Sarah Palin, and in that case, they are not. But I'm not going to go there. These genes are all evolutionarily related. Oops, excuse me. And it's felt there are approximately 80 potassium channels in the human genome, and they're all felt to have arisen from a progenitor potassium channel gene with 6 transmembrane spanning segments, and 4 of these have to tetramerize, come together to form a functional channel in the membrane. So this is like a donut with a hole in the middle. The ions can go through the hole in the middle, except that there are gates that can open and close and stop the ions from flowing. And furthermore, the hole in the middle can regulate the flow of the ions so that only potassium can get through. Sometime during evolution, unless you live in Alaska, there was a duplication of the gene so that there were 2 of them in tandem, and then a reduplication so there were 4 of them, such that sodium channels and calcium channels, of which there are 10 or 11 each, now have a transcript that encodes a protein with 4 of these internally homologous domains in a single polypeptide, so a single protein can form a functional channel. that is now specific for sodium ions or calcium ions. So there are lots and lots of different ion channels expressed in different tissues. And as we had predicted, not only do mutations in these kinds of channels cause periodic paralysis and non-dystrophic myotonia, but they also, in different channels, cause episodic ataxia, epilepsy, familial hemiplegic migraine, long QT syndrome. So the prediction we made in the cloning of the first channelopathy gene all came true. All these other organ systems can be affected, and the symptoms that are manifest depend simply on where the channel is expressed. So in the case of all of the top 6 of these diagnoses, everything but Anderson-Tawil syndrome, all of these, the genes are expressed exclusively in skeletal muscle, and so the phenotype affects skeletal muscle only, and there's no affection of other tissues. Anderson-Tawil syndrome I want to spend a little extra time talking about because the gene is much more widely expressed and the phenotype is much more pleiotropic. We know, for example, that there are cardiac arrhythmias in Anderson-Tawil syndrome, so as you would predict, the gene, and as we have shown, the gene, the causative gene KCNJ2 is expressed not only in skeletal muscle, where you would expect it to have periodic paralysis, but also in cardiac muscle, which gives rise to these cardiac arrhythmias. Furthermore, the original description in a single case by Anderson described a developmental phenotype of the distal limbs and of the head and face in structures that arise from neural crest. And as predicted, we've shown KCNJ2 is also expressed in neural crest. We've also gone on to show that patients with Anderson-Tawil syndrome have a dental phenotype. And I was reminded, although I hadn't remembered, that part of tooth development comes from neural crest. And they also have a neurocognitive phenotype, as we've shown, which is really interesting because KCNJ2 is most highly expressed in the frontal lobe. So they have difficulty with frontal executive function and with other neurocognitive tasks. It's a very specific phenotype. Bruce Miller and Joel Kramer assure me. And so this is quite different than the other periodic paralyses which affect only muscle. But that is not unexpected given that this gene is much more widely expressed than in just skeletal muscle. And so all of these muscle diseases now can be recategorized molecularly based on the genes and the mutations that cause them. Now, I don't have time to show you all the data, but we've done extensive characterization of the mutations in vitro using cellular electrophysiology. And so we know a lot about the disease and how these physiological changes tie in a way that makes a lot of sense based on what we know about neurophysiology. And this has all led to stratification of patient populations in clinical trials that we have participated in, but that have been led by my colleagues and collaborators, Birch Griggs and Robbie Tawil at the University of Rochester. So it's not the same, of course, as finding a cure for the disease, which we'd love to do, but we've improved the treatment of these patients based on a better understanding of the genetics and an ability to parse out these patients, not just to throw them all together into a clinical trial, but to parse them out into groups that have mutations in the same gene rather than taking a collection of patients that are similar but actually at a molecular level have different bases for their diseases. Okay, so this is the point Benitez Lighthouse, and a point to remind me now that I'm going to move on to another interesting group of episodic diseases called the paroxysmal dyskinesias. Dyskinesias are hyperkinetic movement disorders, abnormal movements that include choreoathetosis and dystonia. So chorea is small dance-like movements, athetosis is writhing movements, dystonia is involuntary movement into a more fixed position. And so these are movements that we see commonly in Parkinson's disease patients, severe Parkinson's patients who are treated with large doses of dopamine end up getting dyskinesias. We see choreoathetosis in Huntington's disease. We see dystonia in generalized dystonia. These disorders are rare disorders, but we see them definitely in this group of disorders called the paroxysmal dyskinesias. And there are three main phenotypes I'm going to talk about two of them. One is paroxysmal kinesiogenic dyskinesia. The second is paroxysmal non-kinesiogenic dyskinesia, and then a really interesting one called paroxysmal exercise-induced dyskinesia. I'm going to start with the middle one first, PNKD for short, paroxysmal non-kinesiogenic dyskinesia. This is a gentleman from Rome, in a part of a large Roman family. This disorder comes on in childhood, worsens through adolescence. It's autosomal dominant and highly penetrant. Stress brings on these attacks, but so does 100% of the time alcohol and caffeine, coffee and tea. And so this gentleman avoids coffee and tea, and also chocolate sometimes brings these on. For the purpose of this video, he had a double cappuccino. And you can see that he has some small movements, chorea, and some athetosis, and he has some dystonia. The video's a little moving around a little. I apologize for that. Now, I remind the medical students and the residents it's important to put the neurologic exam into a cultural context. Sometimes things that seem to be dyskinesias might just be communication in Italy, so you have to discern between those. There's some dystonia of the right arm. As we focus in on his face here in a moment, you'll see that he is grimacing, and this is facial dystonia. So this disorder affects the face, the trunk, the limbs. It affects the entire body. He's completely cognitively intact, completely normal in between attacks, and this attack will go on for 1 to 4 hours. PNKD. And a very high proportion of these individuals have migraine. And it is my strong clinical impression that migraine is part of this phenotype. This disorder is so rare I can only estimate, but I estimate it's perhaps 1 in 10 million. Migraine, as Dr. Shapiro here will tell you, is exceedingly common. And so it's impossible for us to collect enough of these patients to statistically test and prove that they really are associated. But it is my very, very strong clinical impression that they are. I can't prove that to you. But here are the first 8 families that we collected over practically 20 years. Not a single one from the US is how rare this is. But they come from Italy, kindred 2688, Germany, Poland, Denmark, Belgium, Canada, and Russia, and someplace else. So you can see that it's a highly penetrant dominant phenotype. Shenyang Li, a student in the lab, cloned the gene about 8 years ago, and it's a 13 exon gene shown in the middle here that has at least 3 alternate transcripts, and the one that we think is relevant is the long form shown here. This is CNS specific. All the patients have either an alanine to valine at position 7 or 9, so at the very beginning of the protein, in this first 118 amino acid yellow part, which is completely novel and doesn't have any predicted motifs or known motifs of any sort. The latter part of this protein shares homology with a huge superfamily of proteins called the beta-lactamases, which is a huge superfamily of enzymes that have diverged many different catalytic functions. And this one falls into the glyoxylase family, which is an enzyme subfamily that is important in stress response. This in situ hybridization shows that the gene is expressed in all layers of the cerebral cortex. It's in the hippocampus and the dentate gyrus. It's also in many structures in the limbic system, including the mammillary bodies, the piriform cortex, etc. But it looks like it's primarily in neurons. We have beautiful antibodies for both the C and N terminus, and shown here is our N-terminal antibody labeling neurons. This is a neuronal marker, and they co-localize. This is the substantia nigra and a marker for TH, and it co-localizes with TH. This is the part of the striatum and substantia nigra from which the paroxysmal dyskinesias of Parkinson's are thought to come. Here you can see the Purkinje cells in the cerebellum. It doesn't overlap at all with a marker for oligodendrocytes, myelin basic protein. Here's the cord, the gray matter of the cord, and it doesn't seem to overlap at all with a marker for astrocytes in the spinal cord. So it's a CNS specific, a neuron specific, and a widely expressed neuronal protein. In a heterologous neuronal-like cell line, SHSY5Y cells, it looks to be in a membrane. I'm having trouble finding, there we go, in a membrane sort of pattern, but we've shown that it's not a transmembrane protein. It's a membrane-associated protein, but it is not transmembrane. Okay, so to look at, to study the protein in vivo, the strategy that we took was to make a back transgenic. This is to take a genomic gene in the genomic context with introns and exons, to engineer in the mutations. This back contains 62 kb upstream from the start site, so we expected it to have all the cis regulatory elements for normal expression and splicing. And we made both wild type and mutant transgenic lines. And in the next slide, I'm going to show you on the right a mutant animal, and on the left, litter mate control after they'd been injected 15 minutes earlier with 25 mg per kg of caffeine. And I was not trained in mouse neurology, but I think you'll agree that this mouse has problems, and it looks really dystonic to me. This attack in this mouse will last 1 to 4 hours, just like in the human patients. We can induce similar attacks by injecting ethanol. We can induce similar attacks by handling the mice excessively, which we presume to be stressful to them. And the mutant mice, and the wild type litter mates don't have any movements of the sort. We've also knocked the gene out in mice, and knockout mice have no movements like this, whether injected with ethanol or caffeine. And so this does not appear to be a loss of function or a dominant negative, but rather a gain of function mutation. This mutant mouse that you saw here on the right still has its 2 endogenous wild type mouse alleles plus 1 or 2 copies of this mutant transgene that I showed you on the previous slide. So this is a beautiful tool now for studying pathophysiology in vivo. So Shenyang injected these mice with, on the top, a wild type mouse, and on the bottom, a mutant transgenic animal with caffeine. Oh, and I should mention, wild type transgenic mice, when injected with caffeine and ethanol, look completely normal. So that's a control for copy number. When I say wild type, the same holds for wild type transgenics. On the top is a wild type mouse, and below a mutant transgenic 2 hours after being injected with caffeine. And this is CFOS immunoreactivity, showing which neurons in the brain are lighting up, and therefore as a marker for activation of the neurons after caffeine injection. And interestingly, all the regions of the brain that show neuronal activation as measured by CFOS immunoreactivity are in the striatum, and they are the parts of the basal ganglia that are in the indirect pathway, shown here as circled in red. Now, as you may remember, the direct pathway signals via dopamine D1 receptors and adenosine 1 receptors. The indirect pathway is D2 receptors and adenosine 2A receptors. And what's interesting, Shenyang pointed out, is that alcohol and caffeine are both nonspecific adenosine receptor antagonists. And so he used A1A and A2A specific antagonists, pharmacologic agents that are specific antagonists, to treat these animals and see if he could induce attacks. And he was able to induce attacks in the mutant mice shown in black bars here with A2A receptor antagonist CSC, but not with an A1A receptor antagonist. So this fits with the indirect pathway data. Similarly, he was able to induce attacks with a dopamine D2 receptor agonist, quinperol, but not with a D1 receptor agonist, also fitting with the indirect pathway being relevant to transduction of of these abnormal movements. Now, this doesn't rule out the possibility that the genesis of these abnormal movements are in the cortex, the glutamatergic inputs into the striatum, but they're certainly being transduced, at least in part, through the indirect pathway. And then Ellen Hess, a collaborator from Emory, did in vivo microdialysis, and then in the red bars here showed that the extracellular dopamine levels in mutant mice are lower at basal levels after stress and after caffeine injection. But interestingly, there's a relative increase with stress and caffeine in extracellular dopamine levels. Wild-type mice have higher levels under all conditions, and there's no change with any of the conditions. So that was kind of interesting. Lower dopamine, but a relative increase. And then Shen Yiguo, a former postdoc in the lab, took our antibodies against the PNKD protein and did a pull-down experiment to begin asking questions about the molecular function of PNKD by trying to understand what proteins it interacted with. And when he found this band, which doesn't show up well here, that was present in wild type and mutant transgenic animals, but it was not present in knockout animals, and did mass spectrometry to identify proteins, to sequence proteins, one of the high hits was RIMS2, a synaptic protein. I'm not, given the limited time, I'm not going to go into all the details, but he confirmed this as a real, he validated this as a real interaction using a number of different strategies, and furthermore showed that in a Percol gradient with our N-terminal antibody again, that the PNKD protein sedimented in a fraction that contains all the markers for the synaptic membrane, synaptophysin, PSD95, GluR2, and RIM2. So this sort of makes sense that there's abnormal dopamine levels, there's abnormal dopamine signaling, and these mice have, and the humans have an abnormal movement disorder, that the synapse might be involved. So this sort of made some sense to Yiguo, and so he pushed further. Using the antibodies and immunogold EM, he showed that PNKD protein is not in the membrane, in the synaptic membrane, but it is in close proximity. It's at the synapse, although not in the membrane, and it's both on the pre- and postsynaptic side of the membrane, and is not present in the knockout mice as a control. And then finally, in primary culture of neurons from rats that are loaded with V-glutfluorin, these, under the pH of the vesicles inside the cell, V-glutfluorin does not fluoresce. But when you stimulate, as shown in blue here, now these are cells that have been transfected with vector alone, but they have the V-glutfluorin. When Yiguo stimulates these cells at 10 hertz, shown by this blue bar, he then sees release of the vesicles, and then with the change in pH, the V-glutfluorin now is able to fluoresce, and there's an increase along the x-axis of signal, fluorescent signal, that then decreases as it diffuses in the media. And so this is a measure of vesicle release in cells that have been transfected with vector control. Shown in black are cells that have been transfected with PNKD, are treated with a 300 action potentials at 10 hertz, and you can see that there's a significant decrease in the amount of vesicular release. And now, interestingly, in red, with stimulation, these are cells now treated or transfected with PNKD protein harboring the human mutation. There's loss of this decrease in vesicular release, and it looks no different than vector control transfected cells. So Yiguo has shown in this line of experiments that PNKD does bind to RIM2, a known vesicular protein. He's shown that it regulates vesicular release, and that the mutations, and it decreases vesicular release, or puts a brake on vesicular release, and that the mutations, at least in vitro, lead to loss of this repression of vesicular release. Again, fitting with the idea that this is a hyperexcitable phenotype. So that's where we are. We still have obviously a lot of work to further understand what the PNKD protein is doing, but we've made a lot of progress. We think that with this decrease in repression of exocytosis, with the basal low levels of dopamine, I didn't tell you, but actually D1 and D2 receptors and dopamine transporter are all upregulated in the mutant animals. We think this is a setup then for a feed-forward hyperexcitability when there's stimulation of the system, then there are more receptors. They're sensitized because dopamine levels are low at basal, under basal conditions, and so there's a setup there for hyperexcitability. This is how we're thinking about the problem at the present time. Okay, now I'd like to go back to paroxysmal kinesiogenic dyskinesia and then finish up with a short story that has a great Vermont connection. This is paroxysmal kinesiogenic dyskinesia, PKD. This teenager is going to get up from the couch, and the movement, the initiation of the movement of getting up, leads him to have this choreoathetotic and dystonic movement, which lasts for 10 seconds, maybe 15 seconds. He's completely with it cognitively during this period, and then all of a sudden it's done and he's okay. He can have 300 or 400 of these attacks per day with change in velocity. Initiation of sudden movement is the most potent way to induce these attacks, but change in velocity, going from walking to running, sitting to standing, standing to walking, any of these kinds of things can induce this brief period of dyskinesia. So this is PKD. It's different than PNKD, but similar. Many more attacks, much shorter in their duration. And we had mapped this to chromosome 16 many years ago and had been working for 15 years to try to clone the gene. The region containing this gene has 200 candidate genes in it, and we'd sequenced almost every one of them, and we're getting quite discouraged. And because of changes in the technology, we recently undertook whole genome sequencing in 6 individuals, one from each of our 6 best families. Now, another interesting thing about this disorder is we'd been studying it because I had collected these families in the early 90s, beginning in the early 90s, with this episodic movement disorder. But pretty quickly we recognized that if you took a history from the parents, that everybody with the movement disorder, or most people with the movement disorder, had actually had benign infantile convulsions when they were infants. And so this is clearly a disorder which we now call PKD with infantile convulsions, PKDIC. This is a cross or an intersection between this episodic movement disorder and an epilepsy. And in fact, you could argue that whether it's an episodic movement disorder, a paroxysmal movement disorder, or epilepsy is purely a semantic one, and the epilepsy guys and the movement disorder guys can fight over it. But paroxysmal dyskinesias of the basal ganglia, is it epilepsy of the basal ganglia, or is it a movement disorder of the cortex? It really is semantic and largely based on the anatomy, I would argue, and we can argue about that later if you'd like to do that. Well, this is the paper reporting the gene. I just emphasize that this is a huge international collaborative group that we put together that when we published this gene earlier this year, late last year, represents patients and collaborators from 5 continents and 12 countries. All the red underlined individuals are clinicians who have submitted patients. I've seen some of these patients myself, but we have over 100 families now, and this has really been a rewarding and fun project because of this, all of these great collaborators. So let me show you some of the families. I'm not going to dwell on the phenotype. Some patients have benign infantile convulsions without the movement disorder. Some have movement disorder without the epilepsy. Many of them have both. But these are large families from all over the world. They're from African descent, Northern European, Southern European, South American, Asian, Turkish, Indian, et cetera, et cetera. So it is a disorder that appears to affect all ethnicities. So these are the families in which we found mutations. And I'm not showing you any of the details because of the time, but the gene encodes a protein called PRRT. It's a proline-rich transmembrane protein about which absolutely nothing is known. And the 4 alleles that we initially found, we now have a paper reporting an additional 30 families or so, has it more alleles. But they're all truncating alleles, here, 2 nonsense mutations, and then 2 frame shift mutations here and here. And here are those 2 transmembrane domains at the very C-terminus of the protein. Now, the only paper published that mentions PRRT2 is by a proteomics group from Germany that back in 2005 had done a high throughput yeast-2 hybrid screen. And they reported that in this yeast-2 hybrid screen that PRRT2 interacts with SNAP25, which was quite interesting to us because of everything we know about PNKD now being a synaptic protein, a novel synaptic protein regulating exocytosis and mutations therein leading to dopamine dysregulation, dopamine signaling dysregulation. That was a yeast-2 hybrid screen, so you're going to find lots of false positives. We've gone on, and again, I'm not going to show you all the details, but we've gone on to validate that this is really a true interaction using a number of complementary strategies. And when we co-localize flag tag or co-express flag tag SNAP25 and HA tagged PRRT2, this is wild type PRRT2, you can see here's HA PRRT2 and here's the SNAP25. Interestingly, when we have mutant PRRT2, it hardly expresses at all. And we've shown now that these mutations cause haploinsufficiency, that they lead to very, very little protein, and we've not yet established whether this is from nonsense-mediated decay or if the protein is actually translated and then degraded very quickly, but we're doing the experiments to test that right now. And here are experiments in primary cultures of neurons done by Jim Moss and Robert Edwards' lab at UCSF. This is flag tag PRRT2 transfected into these neurons, and you can see this beautiful signal. And here's the mutant, and again, almost no signal. So either the protein is made and rapidly degraded, or it's never made at all. It doesn't co-localize with MAP2, which marks dendrites and cell bodies, which is quite interesting. And so this is an axonal protein, and again, this is a brand new story, so I can't tell you more about that. But based on what we know about PNKD and the PNKD protein, and based on the fact that we've now validated the interaction of PRRT2 with SNAP25 and shown that it is expressed in neurons and exclusively in neurons. I guess I don't have the northern blot here, or I'm sorry, the western blot. We've shown that PRRT2 is exclusively expressed in the CNS and exclusively expressed in neurons. We are hypothesizing that this is also, that these 2 related but distinct paroxysmal dyskinesias are going to have a final common pathway of pathophysiology in synaptic dysregulation. Okay, so one last example, and then I'll close. So Dr. Fu tomorrow will talk about another whole exciting group of phenotypes that she's done a tremendous amount of work on and that I've been very interested in too, that affect the circadian system and the sleep homeostat. And this is an example of one family from here in Vermont that Bob Shapiro studies, or has studied and phenotyped. And he had sent to us because this family, the affected people in this family get up really, really early in the morning, and they go to sleep really early at night. So this isn't a disease. This is a circadian phenotype. They're early birds. They're morning larks. So if they had their druthers, they would go to bed at 6 in the evening and wake up at 4 in the morning and do that without an alarm clock or coffee or light therapy or anything else. But the real reason that Bob collected this family originally, and in addition to the fact that they have this interesting sleep phenotype, is that they actually have also all have migraine or migraine with aura. And so that was quite interesting. So we had published the paper describing the gene that causes this circadian phenotype, and you'll hear about that tomorrow. But it was so complicated. It had so much data in it. It had human genetics, in vitro biochemistry, a Drosophila model of the mutation, a mouse model of the mutation, a knockout of the gene in mouse. And so we just left the asthma and migraine with aura out completely so as not to complicate the story. But we've gone on now to study the mice along with Andy Charles at UCLA and Casey Brennan, who is in his lab, and have asked the question, is migraine with aura caused by this mutation? Now, we can't phenotype mice for headache. You can't ask mice if they have a headache, obviously, and so we're kind of stuck. But there are lots of other things we can look at. Migraine is not just about headache. Migraine is a constellation of symptoms, not all of them shown here, but it includes aura, can be a visual aura, the physiologic underpinnings being a cortical spreading depression. These patients also have photo and phonophobia. Migraineurs are well known to have normal pain thresholds in between headache attacks, but have very increased pain sensitivity peripheral pain sensitivity during a migraine attack. And so Emily Bates in my lab a few years ago developed a peripheral pain model in a migraine-like situation that we could test in mice. And Andy Charles is very good at many things, but one, measuring cortical spreading depression in living mice. And so we thought if we could measure a couple of different physiological outputs that travel with migraine, that this might help us argue that migraine is actually caused by this mutation. Cortical spreading depression was described by Liao more than 60 years ago, 65 years ago. And what happens if you take a mouse brain, or a rat brain, I think this is, and you stimulate it with a pin or with potassium, you get this excitability that you can see as these squiggly lines with these field recordings. But then following it, you get this period of inexcitability that is this flat line. And you can see that it's spreading across these different electrodes across the cortex are the different channels being recorded here. And you can see that this depression of electrical activity is spreading across the cortex, and the timing of that is at about 3 millimeters per minute. Coincident with that are changes in vascular diameter and blood flow through the vessels. And so this was the first report of the thing that we now know and love as cortical spreading depression. I'm not going to show you, again, because of time, how Andy Charles images it, but they're beautiful movies, and from those he can extract quantitative data about cortical spreading depression. And in the mice carrying the human mutation that causes the circadian phenotype and that segregates with the migraine with aura phenotype in that family, the threshold to induce cortical spreading depression is remarkably lower in mutant animals versus wild-type litter mates. So one piece of physiological data supporting the idea that these mutations might actually be causing, that this mutation might actually be causing migraine with aura. Doesn't prove it, but it helps bolster the case. And then I'm not showing the data, but the mice with the human mutation also on a migraine-like assay in these mice have a much lower threshold for inducing peripheral allodynia, as shown by Emily Bates, a former postdoc in my lab. So a separate measure of something that we know to be part of the migraine constellation, a piece of physiology that travels with migraine headache, even though, as I've said, we can't measure headache in mice. And then in addition, we were worried that, this still doesn't really prove it, but then in addition, we went back to all of our families with this morning lark phenotype, which we call FASPS, for familial advanced sleep phase syndrome. And we took from every family which we hadn't sequenced the CK1 Delta gene, every new family we had collected in the last several years, we sequenced one person from every family, and in this family, kindred 5579, we found a second mutation in the same gene. And remember back when, on the slide where I was saying, how do you prove it's the causative gene? Finding different mutations in the same gene in different families with the same phenotype. This mutation is CK1 Delta. It's a threonine to alanine at position 44. In kindred 5579, 2 amino acids away at position 46, we found a histidine to arginine mutation, 2 amino acids away. So very close by, we found a second mutation in a second family. Casey Hollows in Ingwei's lab did the biochemistry and showed that it had the same in vitro functional consequences as the first mutation. So it has similar biochemical consequences in vitro. It's at a highly conserved residue, 2 amino acids away from the first mutation. The family has FASPS and they have migraine. We went back and Andy Charles, who's a very talented headache doc like Dr. Shapiro, blinded to the genetic data, phenotyped the family, and showed that mutation carriers also have the migraine phenotype. And so these pieces of data all together, co-segregation of the two alleles, T44A and H46R, with FASPS and migraine with aura in the two families, plus the decreased CSD thresholds in the T44A mice and the increased sensitivity or peripheral allodynia, together give us a really strong argument, I think, that these mutations are causative for migraine with aura in the humans. And this paper is currently under revision, so we're going to see ultimately what the reviewers say about this. So this is a project that was started here through the astute work of Dr. Bob Shapiro. I'm going to close now, but our organism of choice using genetics has been the human, but of course I've shown you examples where we can engineer mutations into rodents and where this can be tremendously helpful in studying phenotypes and pathophysiology in vivo. We can also do in vitro studies of many different kinds. But all of these different organisms have lots of things to offer. But ultimately, we want to understand the human brain. We want to use this understanding of normal function and of pathophysiology ultimately to do better drug trials, but then also to develop novel therapies that will help patients with episodic disorders like epilepsy and migraine. I'd like to finish by acknowledging all the folks who have been involved with the paroxysmal dyskinesias and periodic paralysis projects, and finally the folks who have been involved in the headache studies, most notably for you guys, the wonderful Dr. Robert Shapiro here in the Department of Neurology, but also Chris Jones at the University of Utah, Andy Charles at UCLA, and then the folks in my lab and Dr. Fu, my longtime collaborator, and folks in her lab at UCSF. And I'll stop there and be happy to take any questions. Felix.
Question about the cognitive status of the carriers of the PKD mutation. You showed these nice in situs with lots of cortical expression. You showed that it, the normal inhibits transmitter release. So I'm surprised at the clinical phenotype.
There's no recognized cognitive phenotype. Now, having said that, I would be quick to point out that I've never seen one of these patients myself. None of those 8, we have additional families now, but none of those 8 families come from the US. The Russian family, the clinician, when we wanted more blood to be sent for DNA extraction, he was trying to get us to send money, cash, to Russia, and which, you know, these folks, I've asked the clinicians who have sent them, we've lost contact with the Russian clinician. You know, it's hard when you're dealing with families from all over the world, many whom don't speak English, and none of whom I have contact with in the case of PNKD. None of the clinicians that I've asked report any recognized cognitive problems, but it's, you know, in Anderson-Tawil syndrome, there were no recognized cognitive phenotypes. We looked for them because we noticed that expression of KCNJ2 was highest in the frontal lobe, and then we found them. So the caveat there is if you don't look for it, you won't find it. So it would be interesting to look, and one thing we could do, and haven't yet, is to do, you know, some of the, you know, standard learning and memory assays in mice, behavioral assays in mice, which are a lot easier to do than, you know, learning how to speak Ukrainian or Russian and trying to reach that family. But there are no there's nothing so obvious that the clinicians have told us about it. That doesn't mean that there couldn't be interesting and more subtle phenotypes, to be sure.
Will any of the families functional image related to the dopamine system?
Yeah, so Dr. Hamill is asking about functional imaging and the dopamine system. We had a great clinical fellow, Machiko Bruno, who's the first author on a genotype-phenotype correlation paper on PNKD and a really great clinical paper on PKD, who did a joint fellowship with me and Mark Hallett, which really worked out tremendously. And she did get a couple of the PKD folks into NIH for functional imaging. This was not in none of the functional imaging did she use a marker for dopamine system. She was looking in PKD for blood flow changes and you know, for the movement induction, and didn't find anything. So the answer to your question, unfortunately, is no. I think it'd be spectacular to do. You know, again, this is something that would be perhaps more readily done in the mice, just because it's, you know, we don't have to deal with the human subjects. I would love for someone to do that. I'm not going to do that just because we have so many other things to do. If there were a neuroradiology fellow who was really keen to do that, I would love to work with them. And but nobody's done it yet. Yes, Ray.
Mutations in genes that are involved in synaptic vesicle function, right? And yet, when that fellow you showed the film of, when he stood up, it was a very specific distortion of movement that he experienced, and yet it seems like that's something that's more generalized. Why do you think that it's a more selective attack on one specific type of movement? Because it seems like aren't these proteins more broadly expressed than in one specific motor circuit?
Yes, but he can have attacks on both sides of his body. It doesn't always happen. It was much more in his left arm. Is that what you're getting at? Yeah, I don't know why that was in that particular attack. In other attacks, it can affect both sides, or it could affect his right side.
Why is there selectivity, even though it's probably a very broadly expressed synaptic vesicle, involved in chemical signaling in the nervous system? How can you imagine that it might be triggered more selectively like that?
I don't know the answer. We could go back and look at the video. Maybe he activated the left arm more and pushing off of the, it might be that there just isn't a good answer. But one possibility is that he activated systems that were relevant. These are I'm really fascinated by PKD because it's a reflex. So we also in the past have cloned a reflex, an audiogenic epilepsy gene in mice. So these mice seize when you make a loud sound, reproducibly. So that's really cool. You make a loud sound and they seize. patients with epilepsy, some of them will seize if they have if you get a strobe light and turn it on. And many epilepsy patients, even if they don't seize, get photic driving on their EEG, which is abnormal, but it's a reflex to this visual input. There are seizure patients reading epilepsy who will seize only when they're reading, and you can show this on video EEG, and it's just really interesting. You know, you'd never wish this on anybody, but it's fascinating. Photic sneeze reflex is something we're interested in. People who walk from a dark room into bright light, 10% of the population will sneeze a stereotyped number of times. How many of you do that? So, and if you ask your relatives, one of your parents probably does it, half your siblings probably do it, half your brothers and sisters probably do it. It's autosomal dominant. We haven't cloned that gene yet. So all of these reflex phenomena are fascinating. And so there's something about the sensory input. And so it could be as he's, and is it proprioception in PKD? I don't know. There are PKD patients who say, I can induce one of my PKD attacks not by moving, but simply by thinking about moving. I just think that is so cool. I mean, again, and the great news about PKD, by the way, which is part of the reason I went into neurology too, that's another story, a story for another day, is that you can cure this disease essentially with Tegretol. Homeopathic doses that are so low you would never be able to control epilepsy, essentially can take a kid like this who's having 300 or 400 attacks per day, has gone from neurologist to neurologist to neurologist. They say you're crazy, kid's got psychological problems. They come to you, you make the right diagnosis, you give them 100 milligrams, 200 milligrams, a really, really small dose of Tegretol. Next day, 0 attacks. They come back 2 weeks to your clinic. They bring baskets of flowers and apples and oranges. And I mean, this is a really satisfying. You get this one right, and I did that as a third-year medical student without the help of my residents, and it was such a warm and fuzzy feeling that I said I can do this. That's part of the reason I went into neurology.
I feel like your theme of episodic disorders separated by these periods of normalcy. But I wonder I think the work is very impressive, but speaking of impressive, that horse is the one who has the same sodium channel defect in all the quarter horses that have hypercapnic periodic paralysis. So the question is that those horses just stand there and all of a sudden they drop or something like that. I guess the question in the horses, they don't need to eat anything special or they don't need the stress part of it, as opposed to the humans who need, like you pointed out, except for the horses that drink alcohol.
So this gentleman is talking about 5% of American quarter horses. Now, I'm not a horse guy, but apparently, people who are into quarter horses run the quarter mile. And one thing I didn't tell you about the periodic paralyses, one of the best ways to induce an attack is to have these guys go out. Patients of mine in Utah, in that family I showed you, were in the Army and went to boot camp and would hike with an 80-pound pack 18 miles, and they would do fine. They have this muscle disease, and they would do fine. And then they'd come back to camp, and they'd lay down their pack, and they'd be sitting on a bench. So rest after exercise is the best way to get an attack in one of these periodic paralysis patients. So they'd be sitting around talking with their buds, and then the drill sergeant said, okay, now I want 50 push-ups. And the guy goes, oh, I can't, I'm getting paralyzed. Drill sergeants didn't like that. So rest after exercise. So the American quarter horses, this mutation occurred, a spontaneous mutation occurred in some sire way back. Impressive was the one that it was traced back to, but where the mutation originally occurred is not known. And so because these animals have myotonia, which is hyperexcitability, which is like isometric exercise, which then can transition into paralysis, they have these huge muscles, and they are really impressive, hence the name. And so people who like American quarter horses say, oh, that's an impressive looking horse, I'm going to buy one of those. And so this mutation was selectively bred in American quarter horses until I cloned this gene, and Eric Hoffman cloned the human gene in parallel with me. and Bob Brown at Harvard. And then Eric cloned the horse gene, and this created a stir in the horse community, because the question is then, are these good horses or are these bad horses? You know, they're beautiful because they have these big muscles if you're into horses. But they're really crappy at running the quarter mile, because if they run and then they lay down, they don't get back up because they become paralyzed. And because of the weight of a horse, they get necrosis of the muscle. So I don't agree with you that they don't have to do anything or don't have to have stress. I think what happens is that if they have exercise of any sort, most of these horses are show horses. The ones with the sodium channel mutation cannot run the quarter mile. They will die because they'll run, they'll rest, they'll lay down, and they will never get up again. So the ones that have been selectively bred have been show horses rather than, you know, actual race horses to run the quarter mile. But I'm impressed that you know about them.
The show horses that don't run, I mean, that's what I was asking. I mean, if they run, the ones that run and then fall down, like you said, but the ones that are just for show, and because that gene for the muscles is right next to the, I guess it's on the same chromosome. So if you clone in the muscle gene, you get your mutated gene that Impressive had. Those, or the reason I said this is because people just told me they don't run, they haven't done anything, they're just standing there, all of a sudden, plop, they're on the ground. So that's why I asked, because I couldn't see what the stress was.
Yeah, but who knows how much activity they had prior to going down. They might have been walking around. You know, horse physiology is different than human physiology, and you know, I've never studied one of those horses, and so I can't speak with authority, but it wouldn't surprise me if we could look back very accurately if before going down those horses actually had a little more exercise than usual. And with an animal of that size, it might not require a whole lot of exercise, i.e. just walking around the pen a few times. And who knows what a horse feels in terms of stress, so. Yes, sir.
Question on the KCN J2 mutations.
Yes.
Sort of looking where the light is. They're also present in the vascular smooth muscle in the cerebral organs and are responsible for potassium-induced changes in blood flow. So I was wondering if some of these cognitive issues may in fact be vascular and not neuronal in origin.
Well, we've had MRI scans on some patients and haven't seen any evidence for vascular disease, if that's what you're referring to.
Vascular responses would be involved in hyperlinear responses, for example. When potassium goes up ever so slightly, it activates these channels in the vascular response to vasodilation.
Yeah, I think that's probably not going on, but I can't say that with certainty. The reason is that, it's interesting stuff, the neurocognitive phenotype, frontal executive function, difficulty with abstract reasoning. Those are not things that, my intuition is that wouldn't result from difficulties in dynamic responses of blood vessel. I mean, I take your point, but my intuition says that's probably not what's going on here. And there are other examples in the Weaver mouse, for example, with GERK2, where there's a developmental phenotype in the cerebellum. It's a different channel, an inward rectifier, but where cerebellar granule cells move across the cortex of the cerebellum but don't dive down into the granule cell layer. So I think my sense is it's a developmental phenotype, although, again, we haven't proven that either. So. Not to my knowledge.
Okay. Do you have one more quick question?
One more last question.
Okay.
I'm curious about the linkage with the circadian rhythm-related genes, and whether it's truly a genetic association or a environmental or behavioral, because what you described was really diurnal behaviors in these patients, and maybe these cues are coming from their experiences from the environment, living in an altered diurnal pattern versus a true genetic association with circadian genes.
You have to see Dr. Fu's talk tomorrow. It's absolutely biological. But it is also true that all behaviors, on top of our biological tendencies toward many different behaviors is piled all kinds of environmental, familial, cultural, psychosocial factors. And that makes the work that she'll talk about really so impressive, I think. The fact that, you can scrape away all of that stuff and actually get at the basic biology is impressive, because behavioral genetics has been almost, not completely, but almost completely unsuccessful. And I think the story she will tell tomorrow shows that an example where it's been remarkably successful. But that's the challenge. Take away coffee, take away alcohol, take away lots of other drugs, take away stress, take away job responsibilities, take away family responsibilities. And how do you dive below all of that and find out what your body is really telling you, when it's really telling you to get up, when it's really telling you to go to sleep. That's a challenge.